So welcome back to the session on amino acids and proteins, and here we will be discussing on proteins. So we have already seen amino acids, the structure of amino acids, the properties and classification of amino acids. So when these amino acids, and already mentioned that proteins are like polymers of amino acids which are linked together by peptide bond, which is a specific kind of covalent bond. Okay, so how peptide bonds are formed? Two amino acids, they get covalently linked by way of the amide bond, or what is referred as a peptide bond. Okay. And this yield a dipeptide. And likewise, when this bonding it continues, it develops the uh, polypeptides and proteins later. Okay, so how this is formed, we can see here. Okay, so this is an amino acid. Let it be the first amino acid. Okay, and of this amino acid, this is the alpha carbon, this is the carboxyl group, this is the side chain and the amino group. Okay, and this is the second amino acid which is going to be part of the dipeptide. Okay, and of the second what you call amino acid, this is the alpha carbon, this is the amino group, and this is the carboxyl group and the side chain. Fine. So when these two amino acids they come together to form a dipeptide, what happens is it is actually the carboxyl group attached to the the first amino, the carboxyl group of the first amino acid, and the amino group of the second amino acid. They undergo the condensation reaction and release of water molecule to form the CONH, or the amide bond, or the peptide bond. Okay, so what is happening here? Actually, the hydroxyl group from the carboxyl, the COOH, and hydrogen from the NH2, it is released as water. Okay, and with the release of water, what happens? It forms CONH, a covalent bond, which is what is referred as a peptide bond. Clear? Now, the next one we will be having is, it will be attached over here. Okay, that is, if there is another amino acid to be attached, it will be attached over here, right? And it will be COOH here. So likewise, a CONH bond will be formed with the next amino acid. So likewise, it will go. So what happens? So this is how it will be. Okay, so this is the first amide bond or peptide bond over here. This is the first amino acid. Okay, and this is the second amino acid, and this is the third one. Okay, it is represented. You can differentiate it with the R1 group. It's the R groups, right? And you can see this is first what you call peptide bond, and this is the second peptide bond. Okay, and this is a tripeptide. Here you can see it is a tetrapeptide. And everywhere when we see, it is like there is an free amino like terminal end and a free carboxyl terminal end. Okay, it is known as N terminal and the carboxy terminal. Okay, whenever we write the sequence of amino acids of a protein, we start with amino terminal end. Okay, so it is a amino terminal end, and then the complete sequence of amino acid and the carboxy terminal end. That is the last one. Right. So this is about the proteins. So when we see the structure, we can see that the proteins are there can be four different levels of protein structure. Okay, proteins, as mentioned, they are polymers of alpha amino acids, especially the L amino acids, and the structure, the complexity may vary from very simple primary structure to complex quaternary structure, and based upon the complexity, four levels of protein structure are being recognized. That is a primary structure wherein the amino acids, it forms linear sequence, and here you can see it is just the linear sequence. Okay, so it is one after the other. The amino acids will be bonded by peptide bonds, and it forms a linear sequence, and that forms the primary structure, right? And secondary structure, what happens is there is a spatial arrangement of protein by twisting, folding of the polypeptide chain. So this is the polypeptide chain, and you can see the polypeptide chain, it is twisted in a helical manner. Okay, so this kind of spatial arrangement is also considered in secondary structure. It is not only, so what happens is a very long sequence of amino acid, it is twisted or coiled to form the secondary structure. That is a simple thing. Okay, now you will be learning it in detail later, but here the tertiary structure, it is a three-dimensional structure of a functional protein. So we can see that most of the uh, proteins, they are functional when they attain the tertiary structure, most of them. Okay, and because what you call during denaturation, it is they lost the proteins lose their structure, the tertiary structure or quaternary structure, and hence the function is also lost. So the function is obviously disconnected with associated with the structure they attain. Okay, and the complex one is the quaternary structure. When we are speaking about a single protein and the structure of a single protein in a polypeptide chain, we are speaking about the primary, secondary, and tertiary. Quaternary structure comes into being when there are more than one polypeptide chain or more than one protein chain, molecule, etc. Okay, so so each chain forms a subunit in that case. Okay, so there are multi polypeptide chains which comes into play for developing a quaternary structure. It is not a single one. Okay. So these you can see here. This is linear sequence of amino acids that form the primary structure. Okay, so it is a linear sequence of amino acids forming the backbone of proteins or the polypeptides that form the primary structure. So all proteins do have a primary structure, but it needing be function at that in that particular form. Okay. Uh, this is the secondary structure, and here in the case of primary structure, when we are speaking, the predominant bonding is actually the peptide bond. Okay, it is CONH bond between the amino acids. Coming to the secondary structure, there is folding and what we call pleated sheet formation or twisted, etc. So for attaining the stability of the particular structure, there is hydrogen bonding as well. So apart from peptide bond, there are still other bonding between molecules in a secondary structure. Intramolecular bonding, other than peptide bond, can be seen in secondary structure. Coming to tertiary structure, it is a little more. That, for example, when we are speaking about the secondary structure, it will be either helical or it will be uh, pleated form. But in tertiary structure, the same chain can have helical in one part and what you call pleated form in another part. Okay, so it is a complicated one. But for the specific here, there is only one single polypeptide or protein chain here. Coming to quaternary structure, you can see this is the light orange color, right? This is one polypeptide. This is another one. Okay, even though these two are the same, but there are two polypeptide chains of the same kind. Okay, and green one, it is a completely different polypeptide chain, and this one and this one, two copies are there of the same. Okay, if this is beta, this is alpha. Okay, so this is actually hemoglobin. You'll be learning it in your physiology classes. So this is about the protein structure. Okay, so we can see the primary structure in details. Okay, how we can differentiate a polypeptide from a protein. So the term protein is usually used for amino acid chain which contains more than 50 amino acids. So even some consider it as like polypeptides. That is polypeptides even if the amino acids number of amino acids in a sequence it is 100, it is considered to be polypeptides. Okay, more than that it is if amino acid number is more, we consider it as a protein. Okay, then, when we speak about the primary structure, each protein has a unique sequence of amino acids, which is determined by the genes contained in DNA. So, the primary structure is usually primary structure contains the amino acid sequence complementary to the codons present on the mRNA, isn't it? So, during the process of translation, it is based upon the sequence of what you call codons. The amino acid sequence it is formed, and it makes up the protein structure. Clear. So it is based upon obviously what is the codons based upon. It is based upon the genes in the DNA. So ultimately, each protein has a unique sequence of amino acids that is based upon the genes contained in DNA. Okay, and the primary structure is largely responsible for its function. Uh, the amino acid sequence it determines the function. If there is any lacking or absence of any amino acid during the manufacture process, what happens? It is going to affect the function later. Okay, so the sequence is very important. The amino acid sequence it is very important. If there is any mistake during the formation of the amino acid chain, or if there is any structural errors, it is going to affect the function. Okay, and amino acid composition of protein determines its physical and chemical properties. For instance, if there are more amount of hydrophobic amino acids, obviously the protein itself do have a will have a hydrophobic nature. So even the shape, the how it reacts with what amount of hydrophobic interactions it is possible, hydrophilic interactions, how much it is possible, in which part of, for example, the protein when it combines with glycoproteins, carbohydrates, it forms glycoproteins, isn't it? And these may be the what you call the proteins which make up or which form the you may have studied it in your cell biology classes, plasma membrane, isn't it? The plasma membrane contains certain pores for the transport of molecules. For instance, there is GLUT, that is glucose transporter molecule is there. There are aquaporins for the transport of water. So these kinds of what you call pores and carrier molecules, they are all made up of proteins basically. And what kind of proteins, I mean amino acids make up that decides the function. Okay, if it is more amount of hydrophobic amino acids, obviously the hydrophobic molecules may not be able to help in the what you call transport of maybe like hydrophilic molecules. Okay, it determines the properties, it determines the physical and chemical properties as well as the function. Okay, the primary sequence of a protein, it determines the function, and the vast majority of genetic diseases are due to abnormalities in the amino acid sequences of proteins. That is, changes associated with primary structure of protein. You may have studied sickle cell anemia. Isn't it? Sickle cell anemia, how it is caused? It is caused because of the change in one amino acid in the beta, like B chain. So just single amino acid, just a single change in single position in the amino acid sequence itself can cause very various kinds of diseases. Okay, and that also it can be hereditary as well. Now, secondary structure when we speak. So we have already mentioned that in primary structure, the predominant bond is the peptide bond, and the peptide bond, it is formed when the amino group of one amino acid combines with carboxyl group of another amino acid, and we have already mentioned it is CONH, isn't it? Right. So this is regarding the primary structure, and regarding the secondary structure, we can see that the conformation of polypeptide chain, or the what we call um, structural twisting or folding. Okay, it is referred as a secondary structure. Okay, the amino acids are located close to each other in this kind of a structure formation, and two types of secondary structures are identified: alpha helix and the beta sheet. Okay, and you can see